All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, molecular basis of inheritance, and the subtopic it is the translation. Keep the NCERT textbook open with you, if possible, the pen and paper. The nitrogen-based sequence or nitrogen-based language converted into code language, that is transcription, DNA to RNA. Code language of mRNA to amino acid language, that is translation. Only transcription occurs in nucleus. Only translation occurs in cytoplasm. Both transcription and translation occurs in chloroplast as well as mitochondria. Transcription, translation both are coupled in case of bacteria. So in translation, polymerization of amino acid according to the information present on mRNA is translation. So, in polypeptide, the structural units, the building blocks are amino acid. So, all amino acid polymerize to form a polypeptide. The order and sequence of amino acids in a polypeptide determined by the sequences of nitrogen bases in mRNA. And the amino acids are held by peptide bond. The carboxyl group of first amino acid, amino group of next amino acid, the bond formed is peptide bond formation. The peptide bond formation require energy. So in first phase itself, amino acids are activated by utilizing ATP in presence of enzyme amino acyl synthetase. The activated amino acid is attached to third end of the tRNA, cognate tRNA, specific tRNA. What do you call charging of tRNA or amino acylation of tRNA? With the charging of tRNA, amino acylation of tRNA, now tRNA is said to be charged tRNA. Now the tRNA with the amino acid at the third end. When two such charged tRNAs brought side by side, peptide bond formation favored energetically. Direct energy is not available. ATP already invested during activation of amino acid that bound to tRNA. So during charging of tRNA itself, the energy locked in case of ester bond, that is utilized in case of peptide bond formation. So that's why peptide bond formation occurs and it is favored energetically. And the catalyst, the ribozyme, 23S rRNA in prokaryote, 28S RNA in case of eukaryote, the catalyst enhance the rate of peptide bond formation. Next, the protein synthesizing factory that include ribosomes. And ribosomes having structural RNA and 80 different types of protein. And some of structural RNA is going to be a catalytic. And in its inactive state, ribosomal subunits present in a free state, that is dissociated state. When associated, it becomes active. Dissociated, it is inactive state. Whenever SSU, that is smaller subunit of ribosome, encounters mRNA, the initiation of the translation, that is protein formation, begins. Next, the ribosome, it can accommodate, largest subunit of ribosome, it can accommodate two amino acids which are close enough for the formation of peptide bond. So ribosome itself acts as a catalyst. How ribosome itself acts as a catalyst? Ribosome having structural RNA, some of structural RNA perform catalytic function, that is peptide bond formation. What is catalytic RNA? 23S rRNA in prokaryote, 28S in case of eukaryote. What do you call ribozyme? That acts as a peptidyl transferase, facilitate peptide bond formation between amino acids. Next, the translation unit start with AUG start codon end with the stop codon UAA, UAG or UGA. So translation unit flanked by start and stop codon. And before start codon, there is UTR untranslated region at 5 end, and after stop codon, UTR untranslated region at 3rd end present, which is not transcribed but useful for effective translation and recognizing the ribosome. It is important. And before UTR, it is the cap. After UTR, it is the tail present. So overall, from 5 end, cap, UTR, AUG. From 3rd end, stop codon, UTR and tail. So in this cap and tail, neither transcribed elements nor going to be translated. And UTR, which are transcribed but not translated. AUG and other sequences of nitrogen bases which are transcribed as well as translated. But stop codon UAA, UAG, UGA, which is transcribed but not going to be translated. Next, the ribosome attached to mRNA at start codon, that is AUG, that code for methionine. So initiator amino acid tRNA, that is methionine tRNA, whose anticodons are complement to codons of mRNA, that is AUG. Presented the first site, that is the P site. Then elongation phase begins. In elongation phase, incoming second amino acid tRNA, whose anticodons are complement to codons of mRNA, comes and occupies at A site, that is right side. So when two such tRNAs with amino acid side by side, peptide bond formation occurs between the first amino acid and the second amino acid. As ribosomes moves by one codon distance on mRNA, there is addition of amino acid one by one, dictated by DNA, represented by mRNA, till exposure of the nonsense codon. So ribosome moves one codon distance every time till exposure of the nonsense codon. The nonsense codon UAA, UAG or UGA recognized by release factors. The release factors are protein factor which bind to stop codon, bring the termination of translation, that is the termination of polypeptide chain. So release factors help in the release of, that is polypeptide, the termination of translation. In case of transcription, that is Rho factor help in the termination of transcription. So Rho factor refers to DNA, whereas release factor refers to RNA as well as translation. That is one thing. Suppose the mRNA having 72 nitrogen bases. So 72 nitrogen bases means it is 24 codons. In polypeptide, how many amino acids are present? 23 amino acids. Because 24 codon, the 24th codon is going to be a stop codon. So in this 23 amino acids are present. Suppose there is a 72 nitrogen bases in mRNA. So how can you calculate the length of the DNA? 72 nitrogen bases on mRNA means in DNA one strand is transcribed. So one strand is going to have 72. The other strand it is going to have 72. So total it is 144 nitrogen bases in DNA. So for 20 nitrogen bases it is having 34. For 144 how much? Like that. Next. If you see the representation of the translation in case of NCERT, if you divide the diagram into A, B, C, D parts, in case of first part it is A, that is the tRNA earlier brought the serine, it making exit from the ribosome. In case of the B, there is a growing polypeptide chain, that is octapeptide. the octapeptide that is having 8 amino acids, 7 peptide bond. The first one it is a glycine and eighth one it is alanine. So this octapeptide. In case of C, that is ninth tRNA that is having valine occupies it is the C site, that is amino acyl site. Now the octapeptide with 8 amino acid present at A site moves to, that is A site. So octapeptide with 8 amino acid at the P site moves to A site, that is the C site, to make peptide bond formation with alanine as well as the valine. So in case of this, it becomes nonapeptide, that is 9 amino acid, 8 peptide bond. Next, in case of D, the 10th tRNA, it is ready with which amino acid, that is ASN, asparagine. So nonapeptide present at A site, that is the C, got shifted to the P site as the moment of ribosome 1 codon distant. So the tRNAs, it makes exit from the ribosome. So nonapeptide present at P site. So 10th tRNA, it brings the asparagine, comes and occupies that it is the C. So to 9th, the growing polypeptide chain with 9 amino acid, and in case of the C site, that is amino acyl site, the 10th amino acid, that is asparagine present. So nonapeptide moves to C site and going to make peptide bond formation with it is asparagine. So valine and asparagine, in between valine and asparagine, peptide bond form. So now at a C site, that is amino acyl site, that is decapeptide, that is with 10 amino acid. Now the first P site, it is empty. Now the decapeptide present at C site moves to P site. Now the C peptide, the C site is exposed with nonsense codon. So the last amino acid, the growing polypeptide chain present at P site, and at A site, the incoming amino acyl tRNAs and exposed with the nonsense codon. So the last amino acid with in the polypeptide, the growing polypeptide released from P site. So for subsequent topics, in case of the YouTube channel, remaining part in case of the playlist, the subscribe and share to the person who are seriously preparing for NEET.